Hello, my lovely students, and we're going to talk through the 2 required practicals that you need for the B1 paper. So the first one is how to use a microscope. And to help me, I have got my trusty microscope here. Now the first thing to remember is that both the eyepiece and the objective lens have got a magnifier in them. So my eyepiece is usually times 10, so that makes it 10 times larger. And then the objective lens I can change so it'll zoom in further. So this one, for example, is times 4. When you combine that with the times 10 lens up here, you get a total of times 40. Okay, I quite like this. It's times and you're multiplying them. It's quite difficult to forget that one. So you're times 10 times 4, a total of times 40. When I'm doing my slide, I'll put my slide here on the stage and hold it in place. I then look through the eyepiece and I move the stage all the way up to the top to start with, and then I move it down until it's in focus. When whatever I'm looking at is in focus, then I can switch my objective lens to zoom in further. So I've just gone from my times 4 to my times 10, times 10, times 10, actually times 100. So I can look down there and then I can make a nice drawing of my cells. In the exams, you're usually asked about something about magnification. Okay, so at the moment I've got times 100. That's quite easy. So I'm looking down here, I'm drawing my cells and I'm measuring them with a ruler, la 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 la, and I say, right, okay, so it looks like when I do all my measurements here, each cell is about 2 millimeters in size. Fabulous. Hang on a minute, 2 millimeters in size, that's quite big. I'd be able to see that. So why can't I? Because it's actually 100 times larger. So instead of being 2 millimeters, which is 2000 micrometers, it's going to be 1/100th of that. So it's going to be 20 micrometers. Now, an easy way to tell if you've got it wrong or if you've got it right is, oh, it's 2 millimeters in size here. That means in real life, it's going to be 200 millimeters. Is a cell 20 centimeters in size? No. Okay, so you are just using the numbers. Even if you can't remember the sum, the exam usually gives it to you, but if you can't remember, use your common sense. Okay, if the cell appears to be 2 millimeters, it's actually going to be 20 micrometers. If it says you measure the cell and it's 18 micrometers, oh, and it's times 100, oh, okay, if it's 18 micrometers in reality, it's going to look like it's 1,800 micrometers when I'm looking at it under my times 100. Okay, so that's your microscopy one to do. Don't forget to write down the sum, because you do get marks for writing down the sum. So the next one to look at is osmosis. And we always look at osmosis with a potato. But before we do this, we need to know the definitions for diffusion, osmosis, active transport. We looked at this in class, remember? And we said diffusion is like someone who's bad at playing darts. They get their darts, they throw them at the dartboard, and they go everywhere. Diffusion's like that. You get your molecules, which are all in one small area, and they spread out and go everywhere. Active transport's the opposite. And again, it's like darts. You put in the time and the effort and the energy, because active transport needs energy. You put in time, effort, and energy, and you get all those darts close together. You're getting them all, treble top, 180, that would be fantastic. So active transport takes energy, and it takes things from a wide area and puts them in a small area, like tidying up or something like that. It takes energy. Whereas diffusion, things just move around and spread out. Now, osmosis looks at what water is doing. So what I've got here, I've got a nice hot cup of water. Take a look at it. And my nice hot cup of water needs something added to it. I'm going to add a tea bag to it. Now we want the tea to diffuse into the water, but what the water's going to do is the water's going to move from an area of high water potential to an area of low water potential, see, no water in my tea bag, area of low water potential, through a semi-permeable membrane, which is what your tea bag is. You can also say it's going from low concentration to high concentration through a semi-permeable membrane, because right now there is a high concentration of tea in the tea bag, but a low concentration of tea in the water. I always think the concentration one is a little bit trickier to wrap your head around because it's a concentration of everything that's not water. Anyway, if I pop my tea bag in it, and I'll just swirl it around a bit to get things started, great. We can see the tea is starting to diffuse into the water, but the water, through osmosis, has entered the tea bag. And I'm going to leave that there because then I can have a nice cup of tea after I've done the osmosis practical. So, here we go, osmosis practical. I've got a potato. We're going to use a potato. And there's 3 key words you need to know. And these are hypertonic, isotonic, hypotonic. Now, if something is hypertonic, when we think of hyper, we might think of hyperactive, which is where you've got lots and lots of energy, lots of movement. I know that there's a lot more to it than that, but when we think of hyperactive, we think of what we can see, which is lots of energy. Hypertonic is where you've got more. When I say tonic, I don't mean the drink tonic water, I mean minerals in the solution. So hypertonic, which is my pink cup here, my hypertonic cup, I've taken some cool boiled water and I've added lots of sugar to it. Right, so let's see if that's worked. If it has, we've just like revolutionized science because, you know, we don't have to sit around waiting for days for our experiment. I really hope this has worked. So let's try our hypotonic one first. So what we had with this one was we had more sugar in the water than we did in the potato. And we've got that semi-permeable membrane, that tea bag it's got to go through. Now, before we take a look at it, let's just look at this. Here's a molecule of sugar, yeah? It's pretty big. Here's a molecule of water. That's pretty small. This can pass through a membrane, but my sugar is going to get stuck. So that means I can't have the sugar moving into the potato. The water should have moved out of the potato. And if the water moves out of the potato, normally we measure the mass of the potato and we see that it's lost mass, it's lost water. So let's see what's happened. There we go. So I've got a potato here. All right. Let's compare that to the hypotonic. Again, the sugar can't move out of the potato into the water, so the water is going to move from the water into the potato. It's moved from an area of high water potential to an area of low water potential. Let's just see if that's worked. So, all right, so I've got my 2 bits of potato and we should see, oh, there you go, look at that. They were the same size, but look how nice and big and fat my hypotonic one is compared to my hypertonic. This one, oh, it's all squidgy and big and nice. This one, it's smaller, it's thinner, and it's a bit tougher because, oh no, oh look at it, it bends. It's had all the water sucked out of it. This one has had water put into it. It doesn't bend as easily and it's full. The cells are what you call turgid. They're not quite bursting at the seams. The cell wall is stopping that. If this happened to our cells, our red blood cells, they'd burst open. So we've got hypotonic. Hypotonic, it's a high water concentration. The cells get bigger. Hypertonic, low water concentration. Look at it, it's bending inwards as well. The cells, they can't quite shrink, but they get flaccid. The cells get smaller. The cell walls stay the same, roughly the same size, but the cells inside them shrink a little bit. They are flaccid. They are, easier to bend, floppy. So there we go. So that's what happens in osmosis. The water moves from an area of high water potential to an area of low water potential, or a high concentration of, for example, sugar, to where there's a low concentration, to make everything the same, okay? Right, if there are any questions, ask me in class. Otherwise, hopefully you've enjoyed this video, and I now have a nice cup of tea to enjoy. Cheers.